Hello and welcome to AFCAM's webinar on IHC ICC Staining techniques using single and multiple labels. Today's principal speaker is Simon Renshaw, Senior Imaging Scientist at AFCAM. Simon completed his Biomedical Sciences degree at the University of Bradford. After this, he completed his BMS1 training in the Department of Histopathology at Addenbrooke's Hospital in Cambridge, UK. This was where he was introduced to using immunohistochemistry and immunofluorescent techniques for diagnostic purposes. Simon joined AFCAM in 2001. Joining Simon today will be Judith, our product manager at AFCAM. Judith completed her molecular biology degree and PhD at the University of Dundee. Upon completing this, Judith moved to the MRC Laboratory of Molecular Biology in Cambridge, and she joined AFCAM in 2011. I will now hand over to Simon, who will start this webinar.
Thank you, Lucy. Hi, everyone. Welcome to this AFCAM webinar entitled IHC and ICC Staining Techniques using Single and Multiple Labels. As the title suggests, I'm going to talk you through all of the important factors and steps that must be considered when performing both single and multiple label immunostaining. Later on in the presentation, we'll be doing a 3-question quiz just for fun, so watch out for that one, folks. Before we begin, it is worth noticing that a lot of the important aspects of designing IHC and ICC experiments were covered in the webinar Optimizing IHC and ICC Results Through Careful Experimental Design. A video of this can be found in the AFCAM blog. Today's webinar focuses more on the practical aspects of performing immunostaining, both with single and multiple labels. However, for completeness, certain key experimental design steps will be reiterated where appropriate. Okay, so we're going to work through and discuss the following in detail. Firstly, there's IHC ICC using a single reporter label, both day 1 and day 2 of the staining protocol. Day 1 will consist of antigen retrieval, both heat-induced and enzymatic, buffers and detergents, blocking buffers, and finally primary antibody incubation. Day 2 will cover quenching endogenous peroxidases, briefly touch on detection systems, discuss whether to use an enzymatic or fluorescent reporter label, and the various chromogens that are available for enzymatic detection. We will then go on to talk about tinctural counterstains before finally discussing mounting. In case you were wondering, I will talk about fluorescent counterstains in the multiple staining section. In the second part of the webinar, we will look at IHC and ICC using multiple reporter labels, by beginning with how fluorescence works before moving on to combinations of fluorescent reporter labels. We will then talk about fluorescence compared to enzyme reporter labels, followed by simultaneous staining, then sequential staining, finishing with some discussion about controls. Firstly, we will go through a generic IHC or ICC protocol suitable for use with a labeled secondary antibody, polymer, or avidin-binding complex. As you will have learned in the past webinar, there are numerous variations on each step depending on your experimental design. I will talk you through each protocol step and, when needed, I will discuss any common experimental design variations. The focus for the last part of the webinar will be the design of experiments using multiple labels. We'll go into these in much more detail, but it's worth just quickly talking through the individual steps in the protocol just to give you an idea of what's to come. First of all, on day 1, any pre-treatments are performed, such as dewaxing of paraffin sections and antigen retrieval. There is then a wash stage, followed by blocking non-specific protein-protein interactions, or in other words, non-specific staining, after which there's another wash. The primary antibody is then incubated on the specimen overnight at 4 degrees centigrade. We then go on to day 2, and the first job is to wash off the primary antibody before blocking any endogenous peroxidases, if indeed you're using a peroxidase enzyme reporter label. After this, it's time to start introducing the detection system and allowing it to incubate before washing it off. If the detection system you're using is ABC-based, you would add the ABC complex at this point, let it incubate and wash it off, but if not, you'd skip this step. Similarly, if you're using an enzyme reporter label, you'd then incubate in chromogen before washing it off, but if not, you'd also skip this step. You'd also block any endogenous phosphatases at this point if you're using a phosphatase enzyme reporter label. Any counterstains would then be applied before washing off the excess. If you're using an enzyme reporter label and chromogen combination that is not alcohol soluble, you'd next incorporate a dehydrate, clear and mount procedure. But once again, if not, you'd also skip this step. Then finally, whatever happens, you're going to want to mount your stained specimen in some way in order to help preserve it before, during and after microscopic analysis. Let's now discuss the first step in the protocol in a little more detail, which is concerned with performing any required specimen pre-treatments, such as dewaxing of tissue sections and antigen retrieval. Antigen, or epitope retrieval, serves to break the methylene bridges formed during aldehyde fixation, allowing antibodies access to certain fixation-sensitive antigens. Antigen retrieval is therefore only necessary on specimens that have undergone aldehyde fixation, and only then if a fixation-sensitive antigen is being demonstrated. All specimens should be on coated slides to help prevent dissociation. Both frozen and paraffin sections usually survive antigen retrieval if mounted on coated slides, unless they are particularly friable, such as adipose tissue. There are two common methods of antigen retrieval: heat-induced, or HIER here for short, which involves the use of a suitable buffer, such as trisodium citrate at pH 6 or EDTA at pH 9. The other is enzymatic, which involves the use of a suitable enzyme solution, such as trypsin, pepsin, pronase, or proteinase K. Both methods can be incorporated into automated systems, either as part of an immunostaining platform or a standalone antigen retrieval vessel, but if done manually, heat-induced antigen retrieval is commonly performed in a pressure cooker or microwave, and enzymatic in a water bath. The following antigen retrieval protocols are generic, concentrating on the above methods using trisodium citrate at pH 6 for heat-induced antigen retrieval and chymotrypsin for enzymatic. However, please remember that there is no universal antigen retrieval solution. For each antigen, the most appropriate antigen retrieval solution, or enzyme, pH, method and duration must be established.